Alright guys, so today we're going to be talking about photosynthesis and how it relates to physical chemistry. We know a decent amount about how it ties into organic chemistry, but where a lot of research and whatnot is still fairly fuzzy on is the initiation, basically, of the process. And that's due to electron excitation. And we do know that electrons get excited in the photosystems of plants, and they go through the electron transport chain. We'll get into that in a little bit. But what we're fuzzy on is like the initiation and the electron transport itself in some aspects. So I'm going to try to basically just elaborate on what is known and kind of break it down, because I know that a lot of textbooks don't really touch upon this aspect of photosynthesis. But just for some background, photosynthesis is how plants sustain themselves. They do this by creating, you guessed it, glucose. Glucose is a carbohydrate, and in this case, your N value is just 6. Now, they sustain themselves through photosynthesis because it's when plants convert solar energy into chemical energy, and in doing so, provide the essential oxygen gas for the rest of the animal kingdom and other life on Earth. Now, plant cells have chloroplasts, and this is where photosynthesis takes place. Now, while any green part of the plant has chloroplasts, the leaves, and was originally petals, but the leaves is where most of the photosynthesis is taking place. And this is because inside of the leaves, there's a tissue called mesophyll tissue, okay, and that's M-E-S-O-P-H-Y, or here, we have it right here. So we have mesophyll, and that's, in this tissue, each mesophyll cell holds about 30 to 40 chloroplasts. Chloroplasts are truly the crucial factor in the existence of photosynthesis. Now, getting into chloroplasts. So this is a chloroplast, and it has 2 membranes. Inside the interior membrane, we have stroma, and stroma is all the dark here. It is a thick, dense liquid, and it provides structural support for the chloroplast and for thylakoids. And thylakoids are what hold the chlorophyll A and B, and this is the compound that actually gives chloroplasts their green color. Now, the light energy that gets absorbed by chlorophyll allows photosynthesis to occur, and this light energy is what's actually exciting electrons to begin the whole process. So, we know that you have chlorophyll A and B. And so here's your visible light spectrum, right? Roy G. Biv, red, orange, yellow, green, blue, violet. We know that chlorophyll A, and we'll look at a better graph in a little bit. There's the majority of absorbance for chlorophyll A in the red and the blue-violet spectrum, or blue-violet and red sections of the visible spectrum. Now, because this is what's getting absorbed, right? Yellow and green are what's getting reflected, and this is why plants look green to begin with. All the chlorophyll is just reflecting green light back out into the atmosphere. And so we're going to get into how light really plays a role here. So actually, you know what? We're going to go back to the graph for a little bit. So ignore all the Sharpie on the back, sorry. This is your visible spectrum, again, Roy G. Biv. This is wavelength, lambda, in nanometers. And this first line right here that I am sharpening over, that is for chlorophyll A. And chlorophyll A is the main participant in photosynthesis. And again, we're noting that the maxes are at the red-orange section and the blue-violet section of the visible spectrum. And now, in blue-violet, you have what is these photons, which are basically just light particles, we can say. They have the highest energy. Now in the red, they have the lowest energy. And the main distinction here that is pretty essential is more in regards to marine plants. Because we know, so here's some water, right? Water is blue. Big bodies of water appear blue. And so what does that mean? Referring to earlier how we understand that plants are green because they're reflecting green light. Water is blue because it's reflecting blue light, right? So red light is actually the wavelength that's mainly able to penetrate into water, and that's what provides marine plants the ability to carry out photosynthesis and sustain themselves. But because of this, because they're not able to get a lot of the blue light and only red, marine plants tend to be smaller because of this on average. And in comparison to terrestrial plants, they have a huge barrier known as water, or the ocean, that gets in the way of gas exchange. And so this is another factor in why marine plants tend to be smaller. But getting into the electron energy aspect of this, so energy is equal to Planck's constant times the frequency of the electron or the photon. And the frequency, again, is just, so you have a wavelength, right? Trough to trough. You denote this as lambda. We know that frequency times lambda is equal to the speed of light, right? And the speed of light is a constant. So we know that if we increase the frequency, then we're going to have to decrease the wavelength in order to keep C at this value, and vice versa. If we decrease frequency, then we're going to have to increase the wavelength, and hence they're inversely proportional. Now, what this means is that when we're dealing with blue light, which has a much higher frequency, when we plug that into this equation, we're going to see a much higher energy level. And so with red light, right, we're going to have a much larger wavelength. And so we're going to have a much lower frequency. So this is going to be a lower energy level. And again, this is why the energy of blue light is greater than the energy of red. And now, when we're talking about these energy values, we can think of it almost as like an elevator with a staircase, okay? So you have the elevator, can go up and down, and we're just going to think of this as one version of electron excitation. You have the ground floor, and we call this the ground state. This is where it is most stable. The electron is most stable in its ground state, and n equals 1 is the quantum number, because earlier I showed you that E is equal to Planck's constant times frequency. When we're denoting levels, we actually need n as a multiple to show us the energy levels. And so we refer to that as the quantum number, but this is already hinting at quantization of energy, which means that energy for these electrons it can't just be anywhere, right? It's going to be at multiples, because n will only equal whole integers, 1, 2, 3, so on and so forth. It's not going to be just this linear increase, it's going to be levels. And when we're talking about the energy level of the electron, we think of these as the floors, right? So you got first floor, ground state, second floor is the first excited state. But now what happens is you also have smaller discrete energy levels, and we can think of those as the steps, right? Because steps are still, there's no in between from the first stair to the second stair. No one with a wheelchair could go up, basically, if that makes sense. So you still are taking equal leaps in energy, but these are just smaller ones, and those are the vibrational energy levels. And now this actually ties in, because back to quantization of energy. What scientists have seen is that in some applications that, in this case, definitely not photosynthesis, you'll have incident light, aka the incoming light, and that incoming light has a certain energy value, right? And that, because H is a constant, we know that that energy value is based on the frequency. And we know that this frequency actually turns out to be the same frequency of the electron that gets ejected when this photon hits it, right? Now there's obviously a threshold value that is required of the energy to knock this electron off. But when we're talking about electrons getting knocked off, these are free particles now. This is not in reference to the to photosynthesis, to the excitation of one to the other in this case. But even with photosynthesis, we know that when it hits the first chlorophyll molecule, which excites an electron in the next one, so on and so forth, every single electron that ends up getting excited all has that same frequency. And so what that's telling us is, going back to the Roy G. Biv, there's a red wavelength and a blue-violet wavelength, right, that basically maximize the photosynthesis rate, the rate at which photosynthesis occurs in a plant. And those wavelengths, we relate those again to the frequency through that equation. And if that is something that's kind of difficult to get, we can look at units to make it a little easier. Frequency is in 1/seconds, aka hertz, and wavelength is in meters, depicted in nanometers most of the time because that's where the visible spectrum is, but it's in meters, and we get meters per second, which is the speed of light. And so a speed obviously needs to be a distance per time, aka meters per second. But so there's a threshold energy level, and we'll just say, the energy of photosynthesis, there's the minimum value that photon, that incoming photon needs to have is right here, okay? So we don't see electron excitation at any energy value down below this threshold. But there's certain values of energy based on, again, these wavelengths that promote photosynthesis more. And this is still helping us to highlight and understand that there are discrete energy levels. This is, again, referring to the quantization of energy of photons. But what happens is we know that because the frequency of red light is technically in the light spectrum, that is our lowest energy level for any excitation. This is our threshold right here, the red wavelength. Because we know that at wavelengths longer than red, when we start to leave the visible spectrum, we don't have photosynthesis occurring. Photosynthesis cannot occur with microwaves or anything with a longer wavelength than this red wavelength. So we know that this is our threshold threshold wavelength, which in turn highlights our threshold frequency. And what you'll see is if you crunch the numbers, the frequency here is actually just a multiple of the frequency of the red. And so that's showing us, again, where'd my elevator go? That's showing us that we need either something with this much energy or this much energy, so on and so forth, just to kind of further the analogy before we continue here. And so what we also see through research and through what scientists have kind of looked into is that you can have a beam of like 10 million bajillion electrons and you'll have photosynthesis occur as long as their energy is above the threshold, right? Above this dashed line. But then you can also just hinder the intensity to its lowest possible, to the lowest possible intensity, which would be just one beam of electrons, right? Like one electron. And what we see is that photosynthesis still occurs as long as that energy is above the threshold energy. And so that's also showing us, again, more quantization, because it doesn't matter how many there are, it just matters the energy. What just matters is the energy value of that one electron. And now here we're going to get into the actual process. So this is photosystem 2, also referred to as P680. And so here we have the membrane. This is photosystem 2, okay? And now this is on, again, the thylakoids. So there's a thylakoid membrane. Okay, so continuing on, what we've drawn here now is chlorophyll molecules in the walls of this PS2, and then this is your activator complex. This is a pair of chlorophyll molecules down here, and this is the final excitation within the photosystem too. But we will get to that in a moment. And so you have light coming in, and remember, the frequency at which this incident, the incoming light, hits the electron is the same frequency that this next electron is going to exhibit as it becomes excited. And one way to imagine what happens here, because this excitation is going to lead to the excitation of another one, and so on, and so on, and so on, and it's going to keep going. But the way we can almost imagine this is, let's say that we have a bunch of elevators in a row, right? Because here we have an elevator in the second floor. Jeez, I apologize. Second floor hits one in the first floor, and that goes to the second floor, while the one that hit it initially goes back. So we can almost imagine this as a bunch of elevators with pulleys, right? And actually, you know what? This drawing does not correctly convey that. What happens is, so we have a rope with a hook on it, and this one is connected to an elevator, and then you have another one right next to it with another hook right here. Oh, excuse me. And so on and so forth. Now the easiest way I can explain this is that you have an elevator. When you have the incident light, it's like going to the second floor, right? Because this is going to go up. And so now you have the box here, the elevator here. Now, when this, like I said, very unstable, it wants to go back to the first floor immediately. So as it goes back down, it grabs this hook. And it's going to yank the next electron up. And it's going to excite this one. So this one's going to shoot up. And now this is going to be on the second floor. It's going to latch onto the next elevator so that when it goes down, it brings up the next one. And these are electrons all in their own chlorophyll molecules. So excitation, excitation, so on and so forth, right? Until we hit this activator complex. Now, what happens after that is we see, the final excitation for PS2, at least. And this is going to go into the primary electron acceptor. And so basically what happens is it reaches this primary electron acceptor, and an enzyme will then catalyze a water molecule to split into two hydrogen ions and the solo oxygen atom. And then the electrons are going to be supplied by one of the chlorophylls here. Goes up. And what happens is now it's going to travel through the cytochrome complex into PS1, which is also known as P700. And we will get into that. Okay. So, photosystem 2, right? And we got the P680 complex. Final excitation within PS2 to our primary electron acceptor. And now what's actually going to happen is, like I said, we're going to split a water molecule. It's going to be, let's see, where do we have room? This enzyme is going to split this into as many components as possible, almost. Because we're going to have the two oxygen atoms. I'm sorry, this is, yeah, one oxygen atom, the two hydrogen ions, and two electrons, okay? These H pluses, the hydrogen ions, those are actually going to get released into the interior of the thylakoid. So let's say this is the thylakoid interior. On the other side it's not. So the H plus is going to be floating around over here. And now what's next though is that as the photoexcited electron goes from the acceptor and it goes towards PS1, so we're going to have to draw another one. I just personally feel like drawing it out as I go helps to convey some of these ideas a little better. But that has not been the case for the whole video, so I apologize. So now what's happening is this excited electron still wants to go back to ground state, right? It wants to be on the first floor. So this is our initiation of the electron transport chain. And what ends up happening is the electron is going to go down the chain. And again, because it's going from excited to ground state due to the instability, it's releasing free energy. That's actually what gives us the energy to push these hydrogen ions into the thylakoid interior. And then this is actually helping build a proton gradient across this membrane, right? And so this membrane and this gradient is what we use to make ATP through chemiosmosis. Okay, so primary electron acceptor is then going to, yikes. So the primary electron acceptor is going to push the electron to what's known as PQ, which PQ, plastoquinone, and this is part of the electron transport chain, and it's going to carry the electron to a cytochrome complex. To draw that would take a little too much space, so it pushes that to the cytochrome, complex, which then continues down the electron transport chain to PC. And PC, this plastocyanin. Plastocyanin is a protein that is also part of the electron transport chain, and it transfers the electron between the cytochrome complex and the entry point of PS1. So this is photosystem 1, also called P700. And so what happens there is P700 will take that electron, and we also have more incident light. Creating this excitation chain again. And PC will either provide something for the P700, or more incident light will. Because what can happen is the PC can transfer it to this primary acceptor. And then in a series of more reduction oxidation reactions, the PS1 primary electron acceptor will transfer the electron to a protein called ferredoxin. So if it transfers it to ferredoxin, we don't see NADP+ produced. What happens is we'll actually produce more ATP, because this will go through the cytochrome complex again, and then it will repeat this step. But we can have the primary electron acceptor transfer it to FD over here, and then let me get another paper. So and that transfers to the NADP+ reductase, where NADP+ and one of the hydrogen ions that was in the stroma, or the thylakoid interior, that electron gets transferred to allow NADPH to form. Because what you see, you have this hydrogen ion has a plus because it's lacking an electron. The NADP+ is also a plus because it's lacking an electron. And the electron provided here allows those two to bond and create NADPH. So that's basically how the excitation of electrons helps to initiate that first step in photosynthesis, where we create NADPH and ATP. And yeah, that's the gist of the least understood part of photosynthesis in terms of physical chemistry. At least we know how these reactions work, but in regards to these excitation steps, that's where most of the gray area is in photosynthesis.